Okay. This is a more difficult lecture. I have to draw out some recombinations. So if I have to, I will unleash the other half of my brain. All right. Let's start. Okay, so the topic is going to be structure of yeast plasmids, which are designed a little bit differently than bacterial plasmids. So we're going to talk about some of the differences and talk about what makes them similar. So first we'll just talk about yeast. So yeast is the model eukaryote. But why?
It's pretty simple.
It's simple. That's good. What else? What else do you guys think? It's fast. It replicates fast. So because it's simple, for a long time now, we have had sequenced the yeast genome. So in case you don't know, a genome is all the genes in an organism, and it's assembled on the chromosomes. So we have had for a long time the yeast genome. Okay, so that's an important characteristic of model organisms. Since we have had the yeast genome, and we have also had a robust ability to manipulate DNA in yeast, because recombination works really well. So in yeast, there are also what are called many libraries. So we talked about the concept of libraries last time in bacterial plasmids, where you can have plasmid libraries, but you can also have libraries of yeast. Okay, so some of the libraries that we have is a deletion library. What do you think that is?
They've taken out certain genes.
Yeah. So since we know the entire yeast genome, it's been studied so much that there is an entire library of what is basically all the genes individually knocked out. Now, there are some exceptions. If the gene is essential, can you make a knockout? No. So sometimes you can make what's called like a hypomorph, which means you have like a little loss of function. But if it's an essential gene, you can't knock it out. But for the most part, for every gene that exists in yeast, you can find a strain of it from a library that has that gene knocked out. So we have robust, quick ability to study genes. If we want to study some particular pathway, we can just pull a strain from the library that has a gene knocked out, and we can see what happens in the context of having a knockout. Okay, so there's what's called a deletion library, which is knockouts of individual genes. There's a GFP library. What's that? Right. So oftentimes we want to look and see where a particular protein is in the cell. So for yeast, for all about 5,000 genes or ORFs, we have 5,000 corresponding strains. I'm saying this approximately because there are some things that could cause issues, but approximately for each of those genes, we have a strain that has GFP fused to that protein X. So if you are studying, say, gene Y in yeast, and you think gene Y interacts with protein X, you can pull out the GFP protein X from the library, and you can look at where it's localizing, because we have a strain for every single protein fused with GFP. Does that make sense? Okay. We also have overexpression library. What's that? What would that be?
Where genes like upregulated?
Yes, for every yeast gene, approximately 5,000 genes, we have a plasmid that the yeast can carry that upregulates or overexpresses that gene. Okay? So yeast is a model eukaryote because we have all this infrastructure, all these resources. So imagine you want to study something quick. If you were going to study something in, say, a mosquito, and you wanted to make a deletion, you have to make that deletion yourself. Nobody has made that before usually. Or if you want to make a GFP protein, you got to make that yourself. Or if you want to overexpress something, you got to make it yourself. Most of the stuff in yeast has already been made, and it's organized in these nice libraries that you can just order strains, or you can pull it out from your freezer. So that's why yeast is a model system. Okay. So other things, yeast is a fungi. So again, it's eukaryote. It has a nucleus, the model eukaryote. When we're talking about yeast, Yeast is kind of a broader term, a more broader hierarchy. If you see baker's yeast, that's what you know and love as Saccharomyces cerevisiae. I always spell this wrong. I think it's that. Saccharomyces cerevisiae. There's also fission yeast, which is S. pombe. So if you see yeast, sometimes you'll see papers that are studying quote unquote yeast, and they'll be studying fission yeast or baker's yeast. Baker's yeast is the most common. That's what we use. That's what Dylan's using in our lab right now. Baker's yeast. That's what they use to make beer, bread, and lots of applications in industry. Let's see. Okay, so the other thing about yeast that makes it a model organism, the yeast model, is that do yeast have sex? Yes, they do. Yes, they do. They have quote unquote mating types. Mating types. So they don't have like male, female, but they have A and alpha. And these are all regulated by a key transcription factor. Okay. And A's will only mate with alphas. So you can't have an A mate with an A or an alpha mate with an alpha. A mates with alpha. Okay. And just like any organism that has sex, they make quote unquote gametes, right? So they go through cycles where they become diploid. So after they mate, they become diploid and then they become haploid. Okay. So they go through meiosis. So yeast is a great model for meiosis and mitosis. Anything you want to study at the cellular level that's related to eukaryotes, yeast is a good model for that. Is that clear? Okay. So differences. So let's talk about selection. This is where you start to see the first big difference between like actually using yeast in the lab compared to E. coli. Selection. So what is selection? What are we talking about when we say that?
Like how the organism adapts, like evolves to its environment.
Yes, that's more ecological evolutionary. When we talk about selection in biotechnology, what are we often talking about? You're correct. You're absolutely correct. But there's another sort of side of this, which is like if we talked about this in the context of plasmids, right? If you have plasmid and you inserted gene X, you have to force a bacteria to keep that. Otherwise, it's not going to keep it. And so that would be selection. And in bacteria, we select with what? Antibiotic resistance. Antibiotic cassettes. So in bacteria, if we want bacteria to carry plasmid A, we have to add something to the medium, which is an antibiotic that will kill the bacteria, unless it keeps the plasmid. Okay, right? That makes sense. We talked about that. Yeast is different. That's not how we select in yeast. Okay, so let's talk about how we select in yeast. Okay, so there's two terms that you need to know. Auxotrophy and prototrophy. Okay, auxotrophy means the organism can't make everything it needs. So here's a question. Are humans auxotrophic? Yes, because we actually have to like eat stuff. We have to eat stuff. We can't make everything we need. Prototrophy is you're kind of like self-sufficient. You can make everything you need. So wild yeast, wild type, and I should define this here. If you ever see WT in a paper or a publication, that means wild type. That means you got it from the field or it's like a strain background. It's the wild type. You didn't do anything to it. So wild type yeast can make everything it needs, is self-sufficient. Okay. So in the lab, this is important. In lab, we do not work with wild type yeast. unless you're studying like actual like wild yeast. Like that's not what we use when we do genetic engineering of yeast. We use special strains that have been designed and created to be auxotrophic. Okay, so in the lab, I'm just looking at this. Okay, so now I'm entering into strains. In the lab, we don't use wild yeast. We use special strains that have been designed for study, for like lab work. Okay? So let me just define what a strain is. What's a strain? That's good. A strain implies that there's variation. Variation of what? What's varying? DNA, right? The DNA. So that means like if you have strain X, calling something strain X means that strain X has so and so alleles. Okay? So and so alleles. And what's an allele? An allele is a variant of an ORF or a gene. So like we talked about this, like you can have a gene that codes for blue eyes and brown eyes, or in the fly, red eyes, white eyes. These are both controlled by the same gene, but in the white eye, there's a mutation. So it's a different DNA sequence. DNA sequence is different, does not equal the red-eyed gene which is wild type. So it's one gene, but in different organisms, they have different sequences within that gene. That's an allele. Does that make sense? Okay, so that's what an allele is. So when you say strain X, a strain means it's been defined and its definition literally corresponds to a list of alleles. Okay, so I want to show you, let's see if I get into, am I supposed to get into this yet? Let's get into it now. There's a website for yeast. Google. Hang on. Yeast genome. Actually, I already know it. It's yeastgenome.org. The Saccharomyces genome database. Now let me show you. So I'll search for a strain. I know these strains. So one strain we work with is BY4741. That's the name of a strain. Look, you can look it up. So this is a website you can go to that has all the information you would ever need on yeast, the SGD, Saccharomyces genome database. Here's the strain. So we look this up. Now look, it tells you the genotype. These are the specific alleles that it has. MAT A, HIS3 delta 1, LU2 delta 0, MET 15 delta 0, Uro3 delta 0. Those all mean something. We'll talk about that in a second. Go back to my notes. Okay, so we're talking about a strain. Basically, the way that yeast works is we have mutated genes. This is lab yeast, lab strains. We have mutated genes that are, well, essentially here now you're getting into debate. We've mutated genes that are required for synthesis of amino acids or nucleotides. So one of the things you'll see is a gene called Uro3. What's Uro3 important for? You can just guess. It's a gene that's in the pathway to make uracil. Right. Uracil is an essential component of what? RNA. So if you don't have uracil, can you make RNA? No. Will you survive if you can't make RNA? No, you will die. So if you mutate ura3, the yeast will die, except when we grow them, we add ura3 into the media. So now they can just take it in and they don't need that gene because they can take it up from the media. Okay, another common one is, let's see, His3. What's His3? It's a gene. What is it encode? A protein that does what?
Histidine.
It helps synthesize histidine. Can you survive if you don't have histidine? No, you will die. So what we've done is we've created yeast strains. So let's talk about BY4741. Okay, so go back. Let's go look at that genotype. And let's look at these. MAT A, that's telling you the mating type. So it's basically saying like male or female. It's an A. His3 delta 1, that's a mutation in His3. So this strain cannot make histidine. Lu2, it can't make leucine. Met, what do you think met is? Methionine. So now you know why I made you memorize your amino acids. Uracil, can't make uracil. So this is telling you, if you don't add histidine, leucine, methionine, and uracil into your media, this strain will not grow. Okay, so let me talk about this. How does this relate back to the plasmids? Okay, so if we study BY4741, and we know that, so basically when we grow these things in the lab, we make media that they eat. And in the media, we usually give them all the amino acids. We give them all the amino acids. But when we transform them with a plasmid, so let's say we have a yeast, we have plasmid B, and we want this yeast to carry plasmid B, okay? So what we do on plasmid B is we add a marker that encodes ura3, the proper gene. And in the yeast chromosome, that gene is broken. But this is called complementation. We complement that mutated gene by giving the yeast a plasmid that encodes the normal ura3 gene. Now, if we grow that yeast in what's called dropout media, where we make media that has all amino acids minus uracil, what's going to happen to that yeast? It's going to keep the plasmid because it can't survive on this media unless it keeps the plasmid, which gives it ura3. Does that make sense? So this is a special form of selection. In this case, it's opposite of the bacteria. We are taking something away from the media. In the bacteria, we're putting antibiotics into the media to kill it. In this, we're taking something away, and we're saying you can't survive unless you keep the plasmid. So it's still, you can't survive unless you keep the plasmid, but in this case, we're taking something away. Okay. So this is called auxotrophic selection. Different from antibiotic selection. That's why I define that. Okay. And different plasmids, different yeast plasmids can have different markers. You can have a MET marker, methionine. You can have a ura marker. You can have a His marker. You can have a Lu marker. You can have a trip. What's trip? Tryptophan marker. Okay. And all these things you select for by taking that reagent away from the media. Okay. Okay, so let's talk about this. To make these strains, and I assigned a paper that talks about how they actually make these strains, some of the strains that we study. Okay, so let's talk about how they make the strains. To make the strains, you have to make a mutation. What kinds of mutations can we make? What comes to your head? How would you mutate a gene? What? Okay, you can have a frame shift. That's good. Frame shift would be like loss of one nucleotide, 1A, T, G, or C. That could induce a frame shift. What else? What other kinds of mutations are there?
A knockout.
A knockout. A knockout would be what we would call a deletion. Deletion. That means there's nothing. Well, I mean, it depends on the context. Knockout can mean a couple different things. But in yeast, when we say knockout, it usually means complete loss of function. But if you see delta means like, well, okay, again, all these things it gets complicated. But in general, when we think of knockout, a real knockout is nothing left. Like we actually like removed that sequence from the genome. Okay. And there could also be like point mutations, which would create like a loss of function. You might even still make the protein, but the protein is no longer functional. That could be by like a point mutation. Okay. So what would be something that we worry about in yeast when we work with yeast is what's called a reverting mutation. What would that be? Yeah, these things are living organisms and they are always being selected for. They're growing in populations. And even if you tell them, so let's say you have plasmid B that has a ura marker, and it's got a mutation of the ura cassette on the chromosome. There's actually two ways for that yeast to survive, right? One way is to keep the plasmid. The other way for it to survive is to fix the chromosomal mutation. Okay. So this is why it's important to think about how the mutation was created. This is why I assigned that paper. And you need to understand, because if you're selecting for a plasmid, you might actually not have that plasmid. You might actually have selected for something that fixed the chromosome. So let's say you have ORF. Why am I using ORF? I might as well just use URA3. Let's say you got URA3 and you made a single point mutation that made a premature stop codon tag. So what was supposed to be TAC is now TAG. So you made a stop codon that disrupts the URA protein. All the cell has to do to fix that is make one mutation. Mutate that back to a C. That's very easy. For if you have millions of yeast cells growing in a tube that you're perpetually growing day after day after day, that's very easy for the yeast to do. Okay, so this is a problem, which is what we call reverting mutations that can fix themselves. We don't want that if we're going to work with yeast. So what you saw in the paper that I assigned is they're fixing mutations that would usually sometimes revert. And to make them non-reverting, how do you make them non-reverting?
Completely remove it.
Yeah, you completely remove it. You break it beyond repair. So if you literally remove the sequence, it's gone. They can't get it back. They can't reconstruct the gene from nothing. So that's a non-reverting mutation. We talked about deletion mutations. We talked about the URA3, HIS3, LU2, lyse. We didn't talk about that. What's lyse?
Lysine.
It's a gene. It's not lysine. It's a gene that makes lysine. It's a gene that's in the pathway. I should also say most of these things are pathways where you have enzyme A, enzyme B, enzyme C, D, E. And this takes some chemical, changes it to this, changes it to something else, changes it. And then by the end, you have lysine. So what we're doing is we're disrupting this pathway by knocking out usually something that's in the pathway, which would be like lyse2 or TRP1. Okay? So that's how we make yeast. We talked about how yeast media gives them everything they need unless you're selecting. And that is called dropout media. You can make double dropout media, double dropout. So oftentimes in yeast, we talked about in bacteria, you can select for two plasmids. You can have two different types of plasmids. You can do the same thing in yeast. And all you got to do is make plasmid A have, say, ura, and plasmid B have trip. And then how do you make your media? Yep, you leave out uracil and tryptophan. minus. Then whenever you grow that yeast, it's going to have to keep both those plasmids. Yep.
When you do that, is there a problem with like when the A dies, that B takes up some of their some of their stuff? Or do they give off their amino acid that they code for?
Hang on. I drew this improperly. It should be one yeast cell.
Oh.
Does that clarify? Yeah, that was a mistake from me. Good catch. So we don't necessarily have to worry about that. But yes, so what you said, you pointed out something correct. We would not be able to grow two different yeast cells, A and B, and keep them separate. That wouldn't work. Like they would eventually pick up both. This is what I'm talking about, is growing two plasmids in one yeast cell. That was a good point. Good catch. Okay. Let's look at a plasmid. A yeast plasmid. PS2. Okay. So here's a plasmid. Let's look at the things that are on this plasmid. Let me just make sure. Okay. So what's this? What?
I've heard of ketones. I can't remember.
When you see P, what do you think of? What? Yes, promoter. Promoter. And then what do you think Gal1 is? Where have you heard Gal? What? Galactose. So this is going to be some kind of promoter that is involved with galactose metabolism, right? And actually, I won't make you memorize the circuitry, but the circuitry is if you add glucose, the plasmid, this promoter is off. If you add galactose, what do you think it happens? It turns on. Galactose is milk sugar. It's from milk. F1 origin. What's this? Actually, well, there's three origins here. If we look, we see three cases where we've listed aura. Puck, that's a bacterial origin. So why would there be a bacterial origin in a yeast plasmid? What? No, what do origins do? Origins dictate replication and copy number. So why would there be a bacterial origin in a yeast plasmid?
Maybe the yeast plasmid or the yeast cell wouldn't be able to stop it from making a bunch of copies like over and over.
You're thinking too hard.
Did it come from bacteria?
That's closer to the fact. So, well, it didn't come from a bacteria, but we have to build. Where do we build plasmids?
In bacteria.
In bacteria. Can you build something if it doesn't have an origin? No. So when we build these things, it has a bacterial origin because we first build it in E. coli, then we do a mini prep, and then we transform it into yeast when we're ready to use it. Yeast is the final destination. We don't build stuff in yeast. Well, sometimes we do, but in this case, most of the stuff that we build, we always build it in E. coli first, okay? And we store it in E. coli. And then when we're going to use it, we transform it into yeast, okay? So that's why there's a bacterial origin. Let's talk about the 2 micron origin. So there's also a yeast origin. So yeasts are just like bacteria. You got to put in a sequence that tells them to replicate the plasmid. So let's talk about different origins for yeast plasmids, okay? Because it's the same concept. Plasmid origins are important for regulating what?
Copy number.
Copy number. And we care about copy number. If you wanted to design an overexpression plasmid, would you want the copy number high or low?
High.
High, exactly. If you wanted something to express something that was very toxic, would you want it at high or low copy number? Probably low. I mean, there's going to be cases where you want high or low. Maybe that's a bad example. But there's cases when we want high and there's cases when we want low. And we can change that all by the origin. So let's look at the 3 different origins for yeast plasmids. There's 2 micron. There's what's called a sen. And there's what's called an integration vector. Okay? So 2 micron is actually a wild yeast plasmid, which we have stolen its origin. So this is kind of a version of a shuttle vector that we talked about. So yeast in the wild have their own 2 micron plasmid. And we've taken that plasmid, and the 2 micron plasmid is a high copy number origin. So it's about 60 copies per yeast cell. Sen. Where have you seen CEN in biology? Sen. What does that make you think of on DNA? Cent.
Centromere.
Centromere. What's a centromere?
That's what pulls DNA apart, right?
Centromere.
Meiosis.
Yes. So centromere is basically like the middle of the chromosome. It's a sequence of DNA. It's literally a sequence. And special proteins bind at the centromere, and it regulates the copy number of the chromosome. And how many chromosomes are there usually in a haploid yeast? Haploid yeast. Okay, not how many chromosomes, how many copies of each chromosome in a haploid yeast? One. So a centromere, we've taken the sequence of a yeast centromere and we put it onto the plasmid. So that means when you have a sen plasmid, it's always at 1 to 3 copies. It's low. Okay? And if you really, really want to be definitively sure that you only have 1 copy of a particular sequence, you integrate it into the chromosome. You put it on the chromosome. And the way that you do that is through an integration vector. So the integration vector actually doesn't replicate. It integrates. Does that make sense? So lowest copy number origin would be an integration vector. Middle would be 1 to 3 centromere, sen plasmid. High copy number, 60 copies, is a 2 micron plasmid. Okay, so what this picture is telling us is this a high copy number or low copy number yeast plasmid?
High copy.
It's high copy because it's a 2 micron. 2 micron. Okay, I talked about that. What else is on here? It's a year 3 selection. Why is the ampicillin cassette in there?
You grew it in bacteria.
You grew it in bacteria, so you have to select for it also in bacteria when you're building it. What's psych T? Here's what's this?
That's
Right, the restriction enzyme. So what does that mean this is? What I tell you the word for this is? Multi-cloning site. Many restriction enzymes, this is where you insert gene X. So if you have a promoter right here, and you insert gene X here, what comes after the gene X? Terminator, psych T, psych terminator. Okay, so we can look at this and now we can understand. What's this? What's it telling us? This right here. It's telling us the size. This is the size of the plasmid with all this information in there. Okay, so we now can read and map out yeast plasmids. Okay. This is an origin for it's a virus. It's a bacterial virus origin. So viruses have their own chromosomes, their own genomes. Oftentimes they're circular and they have their own origins. It's a virus origin. I don't actually know why it's on this plasmid, but you're correct that we should be curious about that, and we should look into that. But I don't know the answer to that right now. So you've caught me off guard. But I haven't unleashed the other half of my brain yet. Okay, now I have to because this part's hard. Okay, so let's talk about how we integrate. What happens in an integration? And I just want to make sure we have enough time because I have to do this slow. Okay, so let's draw out, actually, there's a figure for this in the paper I signed. Right here. Okay. So let's walk through this, how you would integrate something onto the yeast chromosome. So the first thing is you still need selection. You still need selective cassette because you have to select for the integration just like you have to select for a plasmid. Same thing, you got to select for it. Okay, so hang on, I'm looking at this. So what they're originally selecting for is this urethra. So they build a plasmid. Oh, I see. I remember what they're doing. So their task is they want to delete lysine, the lys2, right? Because this whole paper is about building strains. And you want to build strains that can't have reverting mutations. So their goal is to delete this gene lys2. Okay, so what do? This is the plasmid they start to build, they start off with. What they do is they take what's called HR, homology region left, homology region right, and they use restriction enzyme cloning to clone this homology region into a plasmid. Okay, does that make sense? Okay, so they make the plasmid, that's step 2, or step 1. Step 1 is they linearize the plasmid. So circular DNA won't recombine as well as linear DNA, okay? Because linear DNA can move around, it's more flexible. So whenever you want to do an integration of something, you usually will linearize. How do you linearize a plasmid? You cut it with a restriction enzyme, okay? So what they did, where did they cut it? They cut it right here. Actually tell you, I shouldn't have drawn so much on there, but B is a restriction enzyme. So K is a restriction enzyme. I have that said here. KBS are restriction sites. K, B, B, S. They cut it right at B, which then linearizes that plasmid. Okay. Now what happens is the homology. Have you guys been taught about homologous recombination, where you get, if you have matching DNA sequences, they can find each other and they can basically intersect with each other, anneal with each other, and then you can get a crossover. You learn about this in like basic meiosis, sex. Like sticky ends. Yeah, it's very much like sticky ends. They have a matching sequence where they anneal together, they cross over, and then they it's called homologous recombination. So whenever you see two matching sequences, so if you see homology regions, you can get a recombination event where what will happen is, let's say this is orange and this is yellow. You can produce things like this, right? You can get like recombinations. Okay. So what's happened here is they've linearized the plasmid. This sequence here matches this sequence right here, right? Right. So you get, what do you call it? And I don't know what you call it. What's the name of it? There's actually a term for this. The Holliday junction. You get a Holliday junction here and here. And it basically then unwinds the plasmid into the chromosome. So here now what we're seeing is, here's chromosome. So after integration, here's chromosome. Here is the original gene that they're trying to knock out. It's not knocked out yet. Here's the start of the plasmid, urethra, the end of the plasmid, and here it's the actually what becomes now the chromosome again. Okay, so now the plasmid has integrated into the chromosome. Okay, so step 2 is to linearize and recombine the plasmid onto the chromosome. But their final task is to knock out lys2, and they have not done that yet. But there's still regions of homology in this. So they add what's called counter selection. So I should write this here. Counter selection. You will see this in many different things in biotechnology. What do you think counter selection is?
You select for the year 3 instead of the last 2.
You select against year 3, or what you would say is you'd call it as you're selecting for a loss of something. So if you're selecting for year 3, you take away uracil. If you're selecting for the loss, not floss, for loss of ura3, you can add a reagent called FOA, and FOA becomes toxic if they have ura3. So they die if they have ura3. So now they want to get rid of ura3. So you first tell them in this paper, and you want to read the legend for this, and you want to try to understand this. It's difficult, but you want to try to work through it. So what they first do is they say, we want you to take in the ura3 right here, and they select for it by leaving out uracil. And then after they select for the integration, they want to pop it out. And so they add FOA, which will kill the yeast unless they lose the ura3. So now it selects for a new recombination. So there's 2 recombinations. The first recombination is when it integrates onto the plasmid. Second recombination is when they pop it out. And the left region here will recombine with the left region here. And what's left over on the chromosome is this. And what pops out is a little plasmid that has ura3 and that lys2 sequence from the genome. And this is just lost. Okay? So then what's left over is this, and this is a complete deletion of lys2. Okay? So I know that is complicated because I have done this in the lab, and every time I do it, I have to draw it out like perpetually again, over and over and over on paper. So I know that it's complicated, but you will do yourself a huge favor if you walk through this paper and try to understand the steps of what they're doing here. Because integrations are very common in biotechnology, especially in yeast. Okay, so we talked about counter selection. We talked about FOA. Yep.
How did the plasmid like latch onto the chromosome initially?
So, I wonder if I can erase. I can't erase this. Let me draw out another example. Well, how does it latch on? Okay, that's a good question. Let's just draw it out. 5, 3, 5, 3. Let's draw a matching homology region. 5. Say these are matching. 3, 5, 3. What happens is this decides to come up here. And wait, did I draw the binding to the right one? This is 3 to 5. Yes, so it'll bind here. This is what's called the Holliday junction. And then what's going to happen is this one is now going to go like that. So now what you have is you have tanglement of 2 chromosomes or 2 sequences of DNA that are homologous. This is what's called the Holliday junction. This is how it physically happens. And then there's enzymes that come along. Let's say a nuclease that will cut right here. Oh, shit. I didn't want to do that. They'll cut it right here. And then basically what you get is you get a seal here and a seal here. And so what's left over is tell me if I'm getting this wrong, is this. See? And now we have recombined this and here, and they're different mixing molecules. So if you do this with a plasmid, And I have a video on this. Watch the video because the video maps out what happens visually with the chromosomes. So I signed that on there. I don't want to pull it up because we don't have enough time. But basically what happens is the chromosome can kind of like, the chromosome kind of like bends to like meet the plasmid at its homologous spots. And then it forms the Holliday junctions. And then you get the recombination. So watch the movie. The movie will address exactly that question. And it's only like a 3-minute movie. So just do yourself a favor and watch it. Okay. Does that help with your question? Like, how does it physically happen? Yeah. It's a Holliday junction. Okay. So there's different strains, different strain backgrounds. I should just talk a little bit more about strains. E. coli have these too. E. coli have the strains too. So if you see like BL21 or top 10, these are strains. And that means they have specific alleles that have convey different functions, or different strains have different uses. Okay. So I'm just conveying that. Let's actually look at the paper a little bit more here in the end. So the other point of this paper is this paper right here. This paper walks you through what they're doing, and it gives you a list of strains. You don't need to memorize that. You don't need to look at this. I crossed out the things that you don't need to know. Most of it's stuff you don't need to know, but I wanted to show you this table. This one. This table is a reference of what these mutations actually are. So if you see Ura3 delta 0, look in this column. Deletion of the ORF? Question mark? Yes. That means Ura3 is completely deleted. MET15, deletion of the ORF? Yes, completely deleted. So if you're ever working with a yeast strain and you go to your SGD, you look at that genotype and you don't know what Ura3 delta 0 is, it's your job to look that up, okay? Because you want to know, could that revert back? That could mess up your plasma. That could mess up your experiment. So you want to know that kind of information. And if you read the literature, or you could even just Google Ura3 delta 0, and there's actually like a database, a list that's connected to SGD that will tell you what these things are. So like, let's see, TRIP delta 6-3, it's a loss of function, but is the ORF deleted? Oh, partially. Oh, my thing's not working. Oh my God. It's done this before. Partially. Okay. Let's talk. I think we're almost done. We have 2 minutes. Let's talk a little bit about, there we go. The stuff that the structures I assigned. Let's look at that. Besides this recombination event, which I wanted you to study. Let's look at this. The other paper was a very, very short paper that I assigned. It's like 2 pages. So you should be able to handle that. It's the creation of generic yeast plasmids. Okay. So P, this means plasmid. And what they're showing you here is they've created a bunch of different plasmids that have a series of plasmids that have either a MET25 promoter, Gal1 promoter, Gal1 promoter, Gal-S promoter, and they put each of these right here, okay? And these, they have SEN versions, and they have 2 micron versions. So what you're seeing in that paper is the creation of a library of plasmids that can be used for many different purposes, that have many different promoters, that have many different selective cassettes, and have different origins, but all kind of in the same spots, okay? So look at that paper. It's only 2 pages. Let's see, what else? We went over that. I'll just talk a little bit quick to end with. Common promoter that you'll see all the time in yeast, oh my gosh, is Gal1. The Gal1 promoter is glucose off, galactose on. That's an overexpression promoter. And then that's it. We'll end there. This thing's dying.